Today I'm going to be talking to you about an antiviral system that exists in bacteria and archaea. Both bacteria and archaea are susceptible to attack by bacteriophage, which is a kind of virus, but bacteria cleverly have developed a system called CRISPR to help defend themselves against such attacks. So what does CRISPR stand for? CRISPR is a bit of a mouthful. It stands for Clustered Regularly Interspaced Short Palindromic Repeats. Now this is part of the CRISPR system, these repeats, but it is certainly not all of it. And it appears that the reason CRISPR came by this name is that back at least 15 years ago, researchers noticed an unusual feature of the bacterial genome, which is these short palindromic repeats. Palindromic meaning that they read the same way forward as they do backwards. And this was an unusual feature of the bacterial genome, so scientists started asking questions about what they meant and whether they represented something important. Well, as it turns out, they certainly have come to mean something. The whole CRISPR system has come to mean something extraordinarily important, not only for bacteria, but for the way that CRISPR can be used for our own ends to design systems for genetic engineering. But to begin with the beginning, let's talk about what happens in bacteria. This CRISPR system essentially enables the bacteria to identify and destroy foreign DNA after it has been memorized. And when I say memorized, I mean that it has to have encountered a bacteriophage once already in order for it to be able to use a CRISPR defense system. We'll be going into that in a lot more detail, but for now, just think of it as a kind of adaptive immunity. The same idea as if you were infected with some kind of virus yourself, your body would know how to recognize that virus if you were ever exposed to it again. So this kind of acquired immunity resides in the bacterial genome in the form of something called a CRISPR array. Now the CRISPR segment of the genome has a lot of different working parts, but the part we're going to zero in on right now is the specific CRISPR array or CRISPR loci. And you can see it right here in this figure. So what it is really is a region where we have repeat, spacer, repeat, spacer, repeat, spacer, repeat. So the repeat is simply that. It is something that pre-existed in the bacterial genome, and it is this short, let's say 20 base pair nucleotide repeat. Now the spacer is a whole different animal. It actually comes from the bacteriophage, because what a bacteriophage does is it injects foreign DNA into its host. And the bacterial response to that foreign DNA coming inside the cell is to cleave some of it. And at this point, we're going to call the segment of the bacteriophage DNA that is cleaved, we're going to call it the Proto Spacer. And there are enzymes within the bacteria that can take that Proto Spacer and actually integrate it into the CRISPR array. So you see the red portion that is being injected by the bacteriophage becomes part of the CRISPR array or the CRISPR locus. This is really only the beginning of the CRISPR system, but it's one of its essential features. So now we're going to go on and look at not only these repeats and spacers, but some of the other features that are important to the CRISPR system. CRISPR really has a whole arsenal. And this arsenal, because it is a sort of war against these bacteriophage, is built up over time. The CRISPR array has, let's say, historical events where it has encountered different bacteriophage, and each time it has encountered a bacteriophage, we have this system, as previously mentioned, where the spacer is taken from the foreign DNA and integrated into the CRISPR locus. Now you can imagine this happens at different times and could happen weeks apart or months apart, or it really just depends, right? It depends on the bacterial population and what kind of viruses they might be encountering. Now there is more to the CRISPR arsenal than just spacers, which we've talked about, and repeats, which we've talked about. A very essential part of the CRISPR system are something called Cas proteins. Cas proteins have primarily endonuclease capabilities. That means they're able to cleave foreign DNA. Now, Cas proteins come in a lot of different flavors, but we're going to focus in particular on Cas9. There are a few different types of CRISPR, and we're not going to get into that level of detail, but just know that Cas9 is especially important in a CRISPR called type 2. And this type 2 CRISPR is the one that was ultimately adapted and modified for genetic engineering purposes. Finally, we have a fourth player. This is called the Proto Spacer adjacent motif, or PAM. This is an incredibly short sequence of DNA, but it is a recognition kind of sequence. So in other words, the bacterial enzymes that are in charge of cutting the DNA, the foreign DNA, and getting it inside the CRISPR loci need to have the PAM there in order to recognize that this is a place where they should actually cut the DNA. So again, there are four major players: the spacers, the palindromic repeats, the Cas proteins, and the Proto Spacer adjacent motif, or PAM. Needless to say, this is a complex system. There are other things as well, but if you understand what all of these are doing, you will basically understand CRISPR. This slide really tells it all about how the whole CRISPR system works. It's what I like to call soup to nuts. And if you really study this slide, it will reveal itself to you, and you can understand what is going on in every single phase or every step of the CRISPR system. So first we're going to focus our attention on this red box, which is what we'll call part 1, acquisition. So in other words, the foreign DNA has to be acquired and inserted into the CRISPR loci. So we've talked about that, but we haven't really talked about how that happens. And if you focus your attention on the Cas genes for a moment, you will see that they're just that. They're genes, and they produce Cas proteins. So the Cas proteins, as mentioned, are endonucleases, and they're basically in charge of cleaving the foreign DNA adjacent to the PAM sequence and integrating it into the CRISPR loci. Cas9 is special. We're going to look at it in part 3 of the CRISPR process because it is essential to the cleavage of the foreign DNA after the bacteria has been infected by a bacteriophage the first time. So now we see how the spacer gets integrated into the loci, but there are a number of other parts to look at in this part 1. The first thing I want to point out is it's not immediately evident from this figure that the bacterial genome is double-stranded. We should know that, but it isn't drawn here in such a way as to look as though all this is double-stranded. And I also want to point out that we're used to, when we're thinking about double-stranded DNA, we're used to thinking about transcription and translation, which is exactly what's going on with the Cas genes. Here, however, at the CRISPR loci, we do have double-stranded DNA that is identified as CRISPR RNA, but the fact of the matter is it's DNA that is ultimately transcribed into RNA, as we see here in this primary transcript, but it is never translated. The same goes for the tracer RNA, which is right here. This is double-stranded DNA that is transcribed into tracer RNA, which we haven't really talked about, but we will in part 2. So what is this tracer RNA? Well, one of the first things you might want to notice is that after it is transcribed as a single-stranded RNA molecule, it actually hybridizes to a CRISPR repeat sequence. And this is a first step towards what we call processing of our CRISPR RNA. But here in part 2, for the moment, we're referring to this as CRRNA biogenesis. Why? Well, because primarily we're at the transcription step where the double-stranded DNA is being transcribed into RNA and where the tracer RNA is coming together with the repeats. You will also hear the term processing, because what has to happen next is this whole extended loci that is now hybridized with tracer RNA must be cleaved into individual segments. And each individual segment, as you can see here, is going to represent a spacer RNA, that was the foreign DNA that was acquired, the repeat or RNA, and the tracer RNA. So it can be a little mysterious, the role of the tracer RNA. But what I have learned, and what I believe to be true, since biology can be a bit of a mystery, is that the primary purpose of the tracer RNA is to act as a scaffold or a handle so that this whole CRISPR RNA can complex with this protein, an endonuclease called Cas9. Cas9 is ultimately what's going to be responsible for cleaving our foreign DNA. But it can only cleave our foreign DNA when a few things happen. The Cas9 complex, and when I say complex, I mean Cas9 in complex with the CRISPR and tracer RNA, must encounter the foreign DNA that it is now able to recognize and destroy. So now I want you to turn your attention to the fact that the bacterial cell may encounter a new viral infection. Now, if that is the same bacteriophage as what infected the cell previously and led to this incorporation event, we now have a system where the bacteria can recognize this viral infection when it happens again, because it is going to do the same thing. It is going to inject its foreign DNA into the cell, and if and when this Cas9 complex with the appropriate CRRNA recognizes that same sequence of DNA, and also notice we have this PAM site here. The PAM site also plays a role in helping Cas9 complex with the foreign DNA. Then Cas9 has a few tricks up its sleeves. It has two domains or two areas of the protein that will cleave this double-stranded foreign DNA. I don't exactly remember the names. It's something like the HUVC domain, but the names of the domains don't really matter. There are two domains that play a role in performing the double-stranded cleavage. And as you can imagine, if you cleave this foreign DNA here, you are well on your way to destroying the whole molecule. So that's how CRISPR works. That is a series of acquisition, CRRNA, transcription, biogenesis, processing, and then ultimately interference. And that's when the whole CRISPR system comes together in order to destroy bacteriophage infection, or prevent, let's say, bacteriophage infection upon re-encounter with the same bacteriophage.